Every single day, the male body produces millions of mature sperm cells. Yet a single one of those cells takes over 2 months to create. We are going to build this flowchart study guide together, step by step. A single sequence of steps provides the cellular framework for diagnosing and understanding male fertility. At the very top of your page, write down your 4 foundational stats: location, the seminiferous tubules; timeline, puberty through old age; hormone, testosterone; and duration, 64 to 74 days. Now let's start phase 1, spermatocytogenesis. Draw spermatogonium A transitioning into spermatogonium B at the top of your flowchart. Spermatogonium B undergoes mitosis to grow and prepare for the upcoming divisions. Draw an arrow pointing down to a new, larger circle. This brings us to our first major milestone. Label this new circle the primary spermatocyte. Inside, write 44 plus XY to show it holds the full diploid chromosome count. Before we move on, pay close attention to the suffixes. Many students fail exams by mixing up gonium, cyte, and ensure your personal flowchart strictly reads primary spermatocyte. This entire first phase serves a single purpose: replicating and staging the maximum genetic payload before the complex splitting begins. Draw 2 branches coming down from that cell. This is the first meiotic division, where the primary spermatocyte splits into 2 secondary spermatocytes. Label these secondary spermatocytes and write in their genetic material, which has now split perfectly in half: 22 plus X in one and 22 plus Y in the other. Next is the second meiotic division. Draw 2 more branches extending down from each secondary cell, giving you 4 new circles at the bottom. Name this row spermatids. Their final haploid genetic payloads are 22X, 22X, 22Y, and 22Y. Right now, these newly formed spermatids are just round, basic cells. They are completely non-motile. The genetic division is done, but without a radical physical redesign, these cells are functionally trapped. That physical redesign is phase 3, spermiogenesis. This is a purely physical maturation process. There is no further cell division here. The static, round cell sheds its excess material, elongates its body, and grows a tail to become motile. Complete your flowchart by drawing tails on your final 4 cells and labeling them spermatozoa. 1 cycle yields exactly 4 mature cells. Now, let's map the real-world failure points. Create a new section on your paper and title it Clinical. First are morphological issues. These occur when there is a physical failure in the shape or motility of the final spermatozoa. This medical illustration shows what those physical failures look like: a normal cell, compared to one with a double head, and another with a tightly coiled tail that prevents it from swimming to fertilize an egg. The second category covers numerical issues. This is a failure in the overall production volume. Combining your biological blueprint with these diagnostic terms, medical professionals can isolate exactly where the 74-day manufacturing line broke down. Put down your pen and compare your page to the master blueprint on screen. You have accurately mapped a complex 74-day biological process, tracking a single diploid cell as it divides into 4 haploid spermatozoa. It's the male side of cellular production. Click the link on screen to map out oogenesis and compare how female physiology manages its own distinct timeline and cellular yield.